Have you ever thought about hunting for a virus? Not with a microscope, but with a supercomputer? Well, today, we're going to dive into how scientists in India are doing exactly that. They're waging a digital war against the Zika virus, all to lay the groundwork for the vaccine of the future.
So here's the situation. The Zika virus, the one infamous for its connection to severe birth defects, is quietly spreading and changing across India. The threat is absolutely real. But the way scientists are fighting back, it's straight out of science fiction. They're hunting this thing down, not in a lab, but on their computer screens.
All right, so why is this digital hunt so incredibly important, especially right now? To get that, we first have to understand the new threat that Zika really poses in India today. What's really wild about Zika's story is how fast it went from being this pretty obscure virus to a global health emergency. For 60 years after it was found in Uganda, it was barely on anyone's radar. Then, boom, 2015, it explodes in Brazil, and we get this devastating link to birth defects. By 2016, it was a full-blown public health emergency, and it already started its journey into India.
And this is the real kicker. Zika isn't just hanging around. It's actively spreading in at least 16 different states across India. And the most alarming part of all of this, we have no specific medical defense against it. There's no universal vaccine, no dedicated antiviral drug, nothing. This is the massive challenge that scientists are racing to solve.
So how in the world do you build a defense against an enemy you can't even see? Well, you start by studying the body's own special forces, the immune system, and how it naturally takes care of intruders.
Your body has this incredible defense system, right? When a virus like Zika gets in, these specialized soldiers called B cells get activated. Think of these B cells as tiny, super efficient factories that start pumping out highly specific weapons called antibodies. These antibodies then go on a search and destroy mission to hunt down the virus, latch onto it, and neutralize it. This whole process creates a protective memory, so you're ready for any future invasions.
Okay, so if antibodies are the weapons, the next big question is, what are they actually aiming for? This brings us to the absolute core of vaccine design, finding the virus's Achilles heel. The answer is something called a B-cell epitope. Now, the easiest way to think about this is like a lock and a key. The surface of a virus is covered in all these unique little shapes. Those are the locks. An antibody is like a key, custom-made to fit perfectly into one of those locks. A vaccine's job is basically to show your immune system a picture of the lock ahead of time so it can start mass-producing the right keys. So finding the right locks is, well, it's everything.
Now, finding these locks used to take years and years of painstaking work in a lab, but we're in a whole new era now. Let's get into the really cool part, how these scientists are hunting for the virus's weak spots using computers.
Whoa, okay, I know. This looks incredibly complicated. But don't worry, you don't need to read every single box. Just think of this flowchart as a digital battle plan. It's the step-by-step strategies scientists use to hunt for the virus's weaknesses, using software to do what used to take years of lab work in just a matter of days. This field is called immunoinformatics, and honestly, it's a total game changer.
Let's break that whole chart down. It's basically a four-step digital hunt. First, they get the genetic blueprint of the Zika strains found in India. Second, they feed that data into a bunch of different prediction algorithms to find potential weak spots, those epitopes we talked about. Third, they filter all those candidates, checking for safety, making sure they won't be toxic or cause allergies, and for novelty, which means our immune systems haven't seen them before. And finally, they run virtual experiments, simulating how their top candidates would actually bind with known antibodies. It's like a test drive, but for a vaccine.
So, they ran the digital hunt. And what did they actually find? This is where it gets really fascinating, because we start to uncover the unique signature of the Zika virus as it's evolving in India.
So, after all that computational detective work, what secrets did the computer models reveal about these Indian strains of the Zika virus?
The hunt was a massive success. The computers found a whole new set of targets on two key parts of the virus. And they found two different types of targets. Some are simple, just a straight line of code, basically. But others are way trickier. They only show up when the virus protein folds into a very specific 3D shape. And most importantly, this work confirmed that the virus is actually evolving differently in different parts of India. And get this, just to show you the power of this approach, on just one of the virus's key proteins, the envelope protein, they identified 12 completely new B-cell epitopes. That's 12 new potential targets for a vaccine that have never been documented before. Just amazing.
Okay, now this is incredible. You can literally see the virus evolving. On the left, you've got a part of the virus from Rajasthan. See that little loop sticking out at the top? Now look over at the virus from Maharashtra on the right. That loop, it's completely gone. This tiny little change at the molecular level is a unique signature. It's the perfect example of why a one-size-fits-all vaccine might not work, and why this kind of local, detailed research is so critical.
So this table kind of pulls all their key finds together. They found new targets that both strains shared, which is perfect for a broad vaccine, but they also found something fascinating. A mutation in the Maharashtra strain actually made it an even better target for our immune system. How wild is that? The virus changed and accidentally put a bigger bullseye on its own back. This is exactly the kind of detail that makes precision vaccine design possible.
All right, so what does this all mean in the real world? This isn't just a cool science experiment for a research paper. All of this digital detective work leads to one ultimate goal: creating the actual blueprint for a vaccine.
So why go through all this trouble to find these tiny molecular shapes? Well, because every single one of these new epitopes they found is a potential ingredient, an essential building block for a future vaccine that's specifically designed to protect against the Zika strains that are actually circulating in India and maybe even beyond.
And hey, don't just take my word for it. The researchers themselves put it perfectly in their paper. They said these findings will help in the development of multi-epitope proteins for diagnostics and vaccinology applications. In other words, this research provides the essential raw materials for the next generation of medical tools to fight this virus.
This all leaves us with a pretty powerful question, doesn't it? In an age where new viruses can pop up and spread across the globe faster than ever, could this kind of fast, precise, digital-first approach be our best hope for staying one step ahead? This research on Zika might just be a preview of how we'll fight the pandemics of the future.